Hello everyone and welcome to this latest Science AAAS webinar, Investigating Cellular Pathways Driving Severe COVID-19, Fresh Insights from Longitudinal Plasma Proteomics. My name is Sean Sanders and I'm Director and Senior Editor for Custom Publishing at Science, as well as the moderator for today's event. COVID-19 has caused more than 1 million deaths worldwide. Extraordinary research efforts are being made at the lab bench and in the clinic, aided by initiatives such as diverted funding, accelerated publication channels, increased collaboration, and open sharing of data. To accelerate research into the pandemic, the COVID-19 Technology Access Framework was set up by Harvard University, the Massachusetts Institute of Technology, and Stanford University to pool resources and ensure that research findings and data are shared rapidly and openly to inform the public health response. Within this framework, researchers from Massachusetts General Hospital recently collaborated with Olink Proteomics to carry out one of the largest longitudinal COVID-19 studies to date. This study aimed to generate new insights into the mechanisms underlying immune responses and disease severity in infected patients by serial measurements of over 1,400 proteins in a cohort of approximately 400 SARS-CoV-2 infected patients and symptomatic controls. Hundreds of plasma proteins were found to be differentially expressed between COVID-19 patients and controls, as well as between those with mild and severe forms of the disease. Together with cellular and RNA expression analyses, the study also enabled a detailed dissection of both systemic and tissue-specific responses to SARS-CoV-2. In this webinar, our speakers will present their work using this rich dataset, which has been made freely available to the scientific community, with the hope that it may form the basis of an early diagnostic of COVID-19 and subsequent clinical intervention. Before we get started, I'd like to share some information for our online viewers. At the top right of your screen, you'll find photographs of today's speakers and a view bio link that you can click on to read more details about their background and research. Also to the right is the Resources tab, where you can find additional information about technologies related to today's discussion and a PDF of the slides. After the speaker presentations, we will have a short Q&A session during which we will address some of the questions submitted by our live online viewers. So if you are joining us live, start thinking about some questions now, and please submit them at any time by clicking the Ask a Question button to the right of the slide window, typing the question into the message box, and then clicking Submit. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the top left of the screen. For tweets, you can add hashtag #ScienceWebinar. Finally, thank you to Olink for sponsoring today's webinar. I'm very pleased to introduce today's speakers to you now. They're going to be sharing the stage, so I'll introduce both of them before handing over control for the presentation. Dr. Michael Philbin is an emergency physician and director of clinical research in the Department of Emergency Medicine at Massachusetts General Hospital in Boston and an associated member of the Broad Institute of MIT and Harvard. His research involves early detection and management of septic shock. Over the past decade, Dr. Philbin has been a key investigator in U.S. National Institutes of Health funded multi-center clinical trials, led sepsis quality improvement efforts, and investigated early sepsis detection strategies. Dr. Arnav Mehta is an oncologist, immunologist, and mathematician who is currently a postdoctoral researcher in Nir Harkonnen and Eric Lander's labs at the Broad Institute of MIT and Harvard, and a clinical fellow in hematology and oncology at the Dana-Farber Cancer Institute Partners Cancer Care. His current interests lie at the intersection of tumor immunology and computational biology, with a focus on building new tools for single-cell genomic studies in tumors. A very warm welcome to you both, and I'll now hand over to you, Dr. Philbin.
Great. Thank you, Sean, for that fantastic introduction, and thank you as well to the folks at Science and Olink for providing this great platform to share our data to such a wide audience. And I'd also like to thank the team who did a tremendous amount of work to make this possible. including our other two PIs on the project, Drs. Goldberg and Huck Cohen. Especially like to thank the sample collection and processing group led by Blair Perry, who were in the clinical areas during the surge enrolling patients, and to Lexi, Kyle, and Bonk, who did so much work supporting data collection and analysis. And I'd especially like to thank Arnaf, my co-presenter, who led all of the analyses that we're going to present. And I thank my lucky stars that he's a mathematician. So we aimed to enroll the most acutely ill patients who presented to the emergency department during the spring 2020 COVID surge, utilizing existing infrastructure that we had in place for sepsis research, and specifically to characterize the plasma proteome in the setting of COVID-19 infection, disease severity, and death. We aimed to explore the relative contribution of cellular and tissue compartments in the body to the plasma proteome. And we consider potential utility of the plasma proteome in clinical practice and in research, and specifically in the areas of outcomes prediction, patient phenotyping, and providing a link to understanding the underlying biology. And probably most importantly, we wanted to provide a rich resource to the global scientific community that will eventually include single-cell RNA sequencing data, viral load data, and other great analyses. So our inclusion criteria were patients who presented to the emergency department, essentially who appeared quite ill and were suspected of having COVID-19 infection, and with specific signs of respiratory compromise. So during about a 2-month period, we enrolled 384 patients, mostly without knowing their COVID-19 status, but 80% who were eventually found to be COVID positive. We defined acuity levels as our primary outcome based on the WHO COVID-19 outcome scale, essentially categorizing patients by those who died within 28 days, those who were intubated but survived out to 28 days, those admitted to the hospital on supplemental oxygen, those admitted without supplemental oxygen, and then those who were discharged from the emergency department without having been admitted to the hospital within 28 days. And about 1/3, or 109 of our patients, we categorized as severe, so being acuity level 1 or 2. And as you can see, 42 of those patients died within 28 days. And the severe cohort essentially comprises all the patients who met criteria for ARDS. So patients with COVID-19 infection compared to those that did not have COVID-19 infection, so red versus blue, included a broad age distribution. And over half of the patients that we enrolled that had COVID-19 were Hispanic. The median symptom duration for our COVID positive patients was 7 days, and those who were infected had a higher BMI than those who are not infected, which is consistent with what's been reported. And despite the differences between COVID positive and negative patients, the acuity distribution between the two groups was actually remarkably similar. And interestingly, absolute lymphocyte count was also similar between the two groups. Inflammatory markers, of course, were quite elevated in COVID positive patients, in particular CRP, which is really a hallmark of this disease, as well as elevated LDH as a marker of tissue injury. And interestingly, despite their younger age, GFR at presentation was significantly higher in the COVID positive patients. So all patients that we enrolled had a day 0 draw. We obtained day 3 and day 7 draws on COVID positive patients still admitted to the hospital at those time points. And after 28 days, we retrospectively collected detailed clinical data and classified outcomes. We obtained measurements on 1,472 proteins using the olink Explore platform. We created linear mixed models for each protein, adjusting for age, demographics, and several key comorbidities. And from these, we obtained differential protein expression of each individual protein for the comparisons of interest. We performed unsupervised clustering and mapped differentially expressed proteins to published single-cell RNA sequencing data sets to infer cell type and tissue of origin of plasma proteins. Now, the proximity extension assay, PEA Olink Explore 1536, is a multiplex platform that came online right about the time we began this project. And Olink was eager to process our samples free of cost to us with the intent of immediately releasing the results publicly. And this dual antibody recognition system with labeled DNA oligonucleotides allows for improved specificity in the setting of multiplexing, and PCR amplification of DNA barcodes increases the sensitivity while really only needing a very small amount of sample volume to measure a large number of proteins simultaneously. So our first linear models use COVID status as the main effect, so COVID positive versus negative, and all of the 384 patients enrolled and using protein expression levels at day 0. So this heat map includes the top 200 differentially expressed proteins between COVID positive and negative patients. Each row represents expression of an individual protein over the entire cohort, and each cell represents the Z-score of protein expression as scaled across the row. And unsupervised clustering, as shown here, shows clear separation of the majority of COVID positive patients from COVID negative patients. You can kind of see the red quadrant in the right upper quadrant of this heat map. And if you look at the bar above, represented by red and blue, red being those that are COVID positive and blue COVID negative. So the vast majority of these patients clustered together. Interestingly, there was a small minority of COVID patients that clustered over on the left-hand side with the COVID negative patients. And these patients tended to be older, and they clearly did not mount the pro-inflammatory response that's kind of prototypical of the COVID positive population. So on the right is a volcano plot that shows the top differentially expressed proteins in COVID positive patients on the right and COVID negative on the left, really highlighting the many changes that we see between the two groups. And in COVID positive, we see a clear antiviral interferon-driven response led by CXCL10, so chemoattractant for a number of inflammatory cells and induced by interferon gamma, which is also highly differentially expressed. DDX58, which is linked to type 1 interferon response and viral sensing, as well as a number of other proteins involved in myeloid and T-cell responses, and a few as well involved in epithelial damage, KRT 18 and 19. And the proteins on the left include NT-proBNP and FGF23 that are associated with CHF, and along with some other proteins on that side, likely reflect the disease presentation of patients who are COVID negative that we enrolled. And this is just an example of a couple of the proteins expressed both in the COVID positive and the COVID negative side, showing differential expression. So we then mapped the proteins that we found differentially expressed between COVID positive and negative patients to published single-cell RNA sequencing expression data in peripheral blood mononuclear cells, so PBMCs, to infer their contribution, potential contribution to the plasma proteome. And again, with relative protein expression scaled across each row, we mostly see enrichment of myeloid cell lines and plasmablasts, and a smaller degree of CD8 positive T cells and NK cell expression. So similarly, we mapped our differentially expressed proteins to published lung single-cell sequencing data. And again, we see an enrichment of the myeloid expression, similar to what we saw in the circulating PBMCs. We also see enrichment of proteins associated with lung epithelial cells that might indicate localized cell or tissue damage. And we also see strong enrichment of the CD8 positive T cell and NK cell populations, and as well as CD4 positive T cell signal that we didn't see at all in circulating PBMCs. So these findings demonstrate the likely contribution of specific tissue cells to the plasma proteome and the ability to deconvolute what we're actually seeing in the plasma. So now I'll shift the focus onto the comparison of severe versus non-severe COVID-19 cases. So the 309 patients who were COVID positive. And again, with severe, including those who died and those who required intubation but survived. So our A1 versus A2 comparison. And essentially, like I said, all of whom met criteria for ARDS. So on the left is unsupervised clustering by UMAP using plasma proteins of our COVID-19 patients at days 0, 3, and 7. And we see a distinct clustering of acuity level and severity in the two upper UMAPs at the top, and considerable overlap with increasing age down in the middle left, with the blacker dots representing the older patients. And at the bottom, there seems to be an overlap with underlying heart disease and kidney disease with severity. And on the right is a correlation matrix where I've got our primary outcome, acuity max, circled to kind of aid you visually. And as you can see, the strongest correlations with maximal acuity are with advanced age, acute kidney dysfunction at day 0, LDH and CRP, along with other expected correlations, such as heart disease, kidney disease, diabetes, smoking status, and a couple other markers. And interestingly, what we did not find associated with severity was body mass index, race, or ethnicity. We also measured neutralizing antibodies and levels which were strongly and inversely correlated with age and with very little association with acuity. So this heat map depicts all of the proteins significantly differentially expressed in severe versus non-severe patients, and these are our so-called severity proteins, with some of the representative proteins listed on the y-axis. So in contrast to our COVID positive versus negative heat map, we see a more heterogeneous clustering of protein expression between the two groups, severe and non-severe, but still distinct subclusters. And the volcano plots on the right demonstrate the expansion of the number of severity associated proteins at day 3 and 7, which is 700 plus proteins, more than half of the olink dictionary, which likely, is a reflection that the disease itself is affecting many different immune processes and organ systems, especially over time. And of note, we did a permutation analysis that showed it's highly unlikely that we get this many differentially expressed proteins by chance alone. So these are a few point plots of interesting proteins that kind of gives us a visual of expression dynamics over time in the setting of severity. And some, such as IL-6, the IL-1 receptors, and IL-15 levels, they start high and they remain high over time. Others, such as TNF, IL-18, and ACE2, start at relatively low levels at day 0 and increase in severity at day 3 and day 7 over time. So similar to the COVID status-associated proteins, the expression in circulating PBMCs of our severity-associated proteins, the single-cell data for PBMCs enrich for our severity-associated proteins, again, in the myeloid cell lines, particularly monocytes and plasmablasts, and to a lesser degree, CD8 positive T cells and NK cells. So our final linear model focuses only on those patients with severe disease, or ARDS, and again, comprising of patients who died versus those that were intubated but survived. And we see differential expression of generally reported cytokines and chemokines, such as IL-6, IL-8, CXCL10, but also relatively novel and less reported, but significant markers, including chemokines that attract monocytes and T cells, so CCL2, CCL7, 8 and 20, IL-1, RL1, receptor for IL-33 that activates T cells and mast cells, and then PTX3, a regulator of innate immunity amongst others. And the typical dynamic over time of these proteins is a kind of a persistent pro-inflammatory response in patients who die, and that's in the red in the point plots, compared to decreased inflammation, particularly at day 7 in patients who survived. We also observed a number of exocrine pancreas proteases and protease inhibitors differentially expressed in those who survived. And we don't entirely understand their relevance, but many have shown anti-inflammatory effects in mouse models, kind of lending to the observation that decreased inflammatory proteome over time is associated with better outcomes. And this depicts Kaplan-Meier curves that demonstrate survival out to 90 days based on day 0 protein levels of some of the selected biomarkers from our model. And some of them really demonstrate stark mortality differences, and again, using day 0 protein levels. So separate from our linear mixed models, we constructed an elastic net logistic regression model with 28-day severity as the outcome, which yielded a fairly good classifier of severity outcome using day 0 protein expression only. We also found that variables selected for this model had high overlap with those that were significant in our linear mixed models, which was validating. And in summary of the models, we observed very distinct antiviral and interferon response to infection in the plasma proteome, with expression enriched in myeloid and plasmablasts and PBMCs, and in particular T cells and NK cells in the lung. And then comparing severe to non-severe, we see heterogeneous yet robust signatures and a number of plasma proteins associated with death. And with this as a basis, I'm going to hand it hand the presentation over to Arnav to talk about specific tissue damage signatures and ligand receptor communication.
Fantastic. Thank you, Mike. So as Mike pointed out early on in the presentation, one of our goals with this was to take the plasma proteome and deconvolute the relative contribution of peripheral cells and tissue cells to the proteins that we see. And not just in the context of COVID-19, our hope is that we could build a resource for the community at large to use this kind of analysis for other disease states. So the intuitive approach that we took was to initially think about the plasma proteins as those that are generally intracellular, and we think of those as cell death markers because the only conceivable way that they would get into the plasma protein, into the plasma would be through cell lysis or cell death. And those proteins that are secreted or membrane bound that might be mediators of cellular communication or signaling. So we first sought to derive a tissue-specific protein signature for every tissue of interest. And to do this, we started with the GTExt RNA-seq data that's publicly available. So here we derived, by looking at the most variable genes that are highly expressed, we derived signatures of genes that are most are tissue-specific for things like lung, kidney, liver, pancreas, amongst other things. Then taking our tissue-specific gene sets, we looked for overlap with markers, plasma proteins that we had looked at in our plasma proteomic data. And we found there was fairly substantial overlap with the olink data set. And in addition, we had access to a somologic data set that also had a poorer dynamic range, but a much more expansive set of proteins. And so using the two data sets, we looked for overlapping markers with each of our tissue-specific gene sets. And then we honed in on intracellular proteins amongst those as markers of organ-specific cellular damage. So here you see validation of some of our tissue-specific protein signatures. So on the top left, you see our heart signature derived from intracellular proteins that are specific gene expression-wise to the heart. And on the left, we correlate with clinically measured troponin. And you see a very nice correlation here that's just statistically significant. We validated some of our other organ-specific tissue signatures with other clinical variables that we had. On the top right, you see a negative correlation with our kidney signature to GFR, as one would expect. On the bottom left, our liver signature correlated well with ALT, which is the most specific marker clinically that we measure for intracellular liver damage. We don't see as sharp of a correlation with total bilirubin, which the primary contributor to in the plasma is red blood cells, and AST and alkaline phosphatase, which both have contributions from other organs or tissues in the body. And finally, on the bottom right, you see correlation of our skeletal muscle signature, which correlates very well with clinically measured CPK. So using these signatures, now we compared the relative expression of these signatures between severe and non-severe patients. So on the top left, you see a box plot showing our heart signature in severe and non-severe patients. And you can see as early as day 0, we see that severe patients express higher levels of intracellular death signatures from heart. And this continues through day 3 and day 7. Our lung signature, in fact, was limited because there weren't as many lung-specific genes, but we did find KRT7 as a fairly lung-specific marker that overlapped with our protein set, and again, we see higher levels of KRT7 in severe patients. And The other striking feature was in skeletal muscle, where we do see much higher levels of a skeletal muscle signature, especially at day 3 and day 7 in our more severe patients. So I showed the validation of our plasma heart signature with troponin at day 0, and we found actually that you could stratify patients on survival by just looking at the heart signature alone at day 0. And in fact, we saw that patients that had higher levels of this heart signature at day 0 fared much more poorly when looking at 90-day overall survival. So the next thing we sought to do is try to identify how much of the plasma proteome is contributed by intracellular death within the lung tissue itself, which is where we see, clinically, we see the striking pathology in severe patients who present with ARDS or eventually have ARDS. And so here, what you're seeing are the set of intracellular proteins that are associated with severity based on the linear mixed models that Mike showed earlier. And what we did was map those proteins onto gene expression data from bronchoalveolar fluid that was publicly available from COVID-19 infected patients. And you see that there's a subset on the bottom right of proteins that are very highly expressed in lung epithelial cells, which are the cell types that have higher expression of ACE2 and TMPRSS2. In addition, there are a subset of proteins that are expressed in T cells, and you see that at the very top, and a few other cell types as well. But the strongest signal appeared to be amongst the intracellular proteins within the lung epithelial cells. So within the lung epithelial cells, we were able to define a cell type specific intracellular death signature by looking at the proteins that were most strongly expressed in each cell type. And we actually found that if you look at the expression of those proteins in plasma, it correlates well with ACE2 expression within those cell types. We did not see a correlation with TMPRSS2, but in some other tissue types, like the heart, we did. But overall, this suggests to us that we could potentially use the plasma proteome as a metric for measuring cell type specific death. Next, we switched our focus to the secreted or membrane-bound proteome in the plasma to ask the question of if we can identify markers of immune pathways and cellular communication in the plasma proteome. So we started by doing an enrichment analysis of the secreted proteins in the plasma proteome and found that they were enriched for NF-kappa B signaling, IL-6 signaling, STAT3 activation, which is downstream of some of the viral sensing pathways, in addition to IL-10 signaling, which has been seen in some other groups. Looking at some of the other pathways, we do see elements of lung damage or lung pathology with enrichment in COPD pathology and cardiac hypertrophy signaling, as well as liver damage. All to say that there are the pathways that we find enriched are consistent with what we see with the clinical phenotypes and what we saw earlier with regards to the immune pathways that might be mediating severity. So we then honed in on the set of ligands that are expressed in the circulating proteome, and we asked ourselves, within the lung tissue itself, what is the contribution of these ligands to cellular communication? So we went back to the single cell RNA sequencing data from bronchoalveolar fluid, and we used an approach called cell phone DB, which analyzes cellular communications by looking at the relative expression of ligands and receptors in different cell types within the single cell RNA-seq data. So the heat map on the left shows the number of ligand receptor interactions that are predicted on the left side at day 0 and on the right side at day 3. And the key point we want to make here is that the number of ligand receptor interactions that are predicted to happen increase dramatically over time. such that to say there's more cellular interaction that might be mediating disease severity. And on the right, what you see is the ratio of ligand receptor interaction between different cell types at day 3 compared to day 0. And so the boxes that are darker brown suggest that there is an increasing evolution of ligand receptor interactions between those cell types. And in particular, the things that we found was that a lot of the severity associated ligands were driving interactions between monocytes and lung epithelial cells, between lung epithelial cells and other lung epithelial cells, as well as T cells and monocytes as well. So as a next step, we honed in on those particular ligand interactions to build a better model of the cellular communication that's driving disease severity. What you see here is on the left side are all the ligands that we saw more highly expressed in the BAL single-cell RNA sequencing data within monocytes. So these are monocyte ligands. And we found that most of the death-associated ligands that Mike showed earlier that were differentially expressed in plasma are most highly expressed in monocytes. And these include things like IL-6, CCL7, CCL8, among other things. The majority of monocyte ligands appear to interact with receptors on T cells and altered things like T cell recruitment, activation, and exhaustion. In addition, there was a subset of monocyte-associated ligands that seemed to interact directly with lung epithelial cells as well. Given that we saw that the monocytes might be driving T cell activation and recruitment, we then focused on the receptors that are expressed on T cells and asked ourselves, are there co-regulated groups of ligands that bind to those receptors? So what you see here is that, again, the majority of signals coming to T cells appear to be coming from ligands on monocytes. And the left heat map here shows the correlation in plasma expression of each of the ligands that are binding to T cell receptors on T cells. And what we found was that there were separate co-regulated groups of ligands that appeared to be most highly expressed in monocytes. And in this one example, you see the cluster that's highlighted with the black box that includes CXCL10, CCL7, CCL2, and IL-6, which are also associated with patient death. There were several other clusters of monocyte-associated ligands as well, including the one at the bottom right. But again, highlighting the role of monocytes in mediating T cell recruitment activation in the disease pathology. Next, we focused on lung epithelial cells themselves, given the number of ligand receptor interactions we observed with those. So on the heat map on the left, what you see are ligands that are expressed on lung epithelial cells. And we found that most ligands on lung epithelial cells bind to receptors on other lung epithelial cells, in addition to T cells as well. So on the heat map on the right, you see towards the bottom, you see a module of receptors expressed on T cells that mediate T cell activation exhaustion, suggesting that there are signals coming from lung epithelial cells that are driving T cell activation and exhaustion. And on the very bottom right, you see a module of proteins that include KIT, MET, EGFR, amongst other things, that drive lung epithelial regeneration and growth factor signaling. So that's to suggest that lung epithelial cells drive their own regeneration and growth factor signaling. And this appears most apparent right around day 3, so with ligands that are associated slightly later in time. You see now, taking a receptor-centric view of receptors on lung epithelial cells, on the right, you see receptors that are expressed on lung epithelial cells. And on the left, we again see a correlation heat map of ligands for those receptors. We again, to reiterate the point, we see the majority of those ligands are actually expressed on lung epithelial cells themselves, and there are co-regulated groups of those ligands, including those involved in regeneration and tissue damage, as well as growth factor signaling, as well as a handful that are part of the IL-6 family, including IL-6, LIF, and OSM. And so just to emphasize, we see that most signals to lung epithelial cells are derived from other lung epithelial cells, and we find two co-regulated modules of proteins in plasma that drive regenerative and tissue damage signals. So all in all, we propose a model, but just to summarize, we find that death and intubation can be predicted on day 0 with secreted proteins. We find that tissue damage signatures, especially the heart signature, can predict outcome. The severity-associated proteins that we find in the plasma are associated with many biological processes, and in particular, we hone in on a communication network between macrophages, epithelial cells, and T cells that might be driving disease severity. And to clarify our model, we believe that the macrophages might be the inciting cell type that initially drives the inflammatory process, and we see several of the death-associated cytokines and chemokines most highly expressed on macrophages. This in turn drives early recruitment and activation of T cells. T cells themselves express activation and exhaustion signals and can lead to direct cellular killing of epithelial cells. Epithelial cells undergo damage, either by nonspecific inflammation of macrophages, but in a temporally delayed fashion express signals of delayed tissue regeneration and growth factor signaling. So overall, what we've sought to do is to dissect the systemic host response to SARS-CoV-2 using plasma proteomics. We found inflammatory signatures that might derive from peripheral blood cells, in particular monocytes, plasmablasts, and some from CD8 T cells and NK cells. We then sought to hone in on intracellular proteins that are found in plasma to derive tissue and cellular death signatures. And in particular, we see a strong heart and skeletal muscle signature. And then by mapping our severity-associated ligands to gene expression data from lung tissue and BAL, we find a cellular communication network that we believe might be driving disease severity, involving the macrophages, T cells, and epithelial cells. So with that, we can take any questions.
Wonderful. Thank you so much, Dr. Mehta, and Dr. Philbin as well. And we are going to move right along to questions submitted by our online viewers. Just a quick reminder to those joining us live that you can still submit your questions by clicking the Ask a Question button to the right of the slide window, typing the question into the message box, and then clicking Submit. So the first question, Dr. Philbin, I'm going to put this to you. This viewer was wondering if you have compared your data set with another olink data set, he says perhaps from Sweden, released earlier this year, and if so, whether there are any significant overlaps that would point to compelling targets or biomarkers.
Yes, I'm not sure exactly which data set they're referring to. There is a population of COVID-infected patients with end-stage kidney disease. I believe the study was out of Sweden. And we just saw this a couple of weeks ago, I believe, up on medRxiv or bioarchives. But we were kind of struck by the amount of overlap seen between their derived severity-associated proteins and ours, and which is validating, is quite validating. Our data set is quite a bit larger, so an appropriate validation set. Now, obviously, part of the explanation for the overlap is the fact that we were measuring the same proteins as well. But even in the subpopulation of patients with end-stage kidney disease, these patients are similar to ours, mounting quite a remarkable inflammatory response with the same kind of monocyte and T cell activation associated proteins, and frankly, several of the death associated proteins that we've also observed in our data set.
Great, thank you, Dr. Philbin. Maybe I'll stick with you for this next question. Did a cytokine storm appear in the severely ill patients that you examined? Did you have that data?
So I might let Arnaf weigh in on this question as well, but cytokine storm, and we certainly observed, the cytokines they're typical of the cytokine storm being IL-6, IL-8, and other cytokines and chemokines that are prototypical of the cytokine storm, and these proteins were definitely more highly expressed in our severe phenotype.
Dr. Mehta?
Yeah, and so we do see, what we think of canonically the cytokines associated with a storm. I would just caution using the term cytokine storm because I think it's loosely defined as well. And I would think of this more as sort of a unique inflammatory signature to COVID patients versus other patients, with parallels to what we describe as cytokine storms in other disease contexts. But I think the most sort of, amongst the proteins that we see that are, one, higher in severe patients, and two, even higher in patients that die versus those that survive, are things that we often see in other disease contexts, like IL-6, for example, and CRS with CAR T-cell therapy, in addition to IL-8, CXCL10, all of which have been previously described.
Let me stay with you, Dr. Mehta. This question is about ACE2, and the viewer says that the increase of ACE2 showed in the dynamic graphs of severe versus non-severe. Where were they measured? Was this also in plasma?
So there are two different contexts in which we show ACE2. So ACE2 is part of the olink plasma proteomic assay. And so we do see increases in ACE2 levels in severe patients compared to non-severe patients over time. So that's an interesting phenomenon that we don't quite have honestly a great explanation for. But the second context in which we show ACE2 levels were when we look at the intracellular death signatures for lung-specific cell types. So there we show a correlation of ACE2 levels to those intracellular signatures that we see in plasma. So in those contexts, we're showing ACE2 levels by gene expression because we're positing that the signatures we see in the plasma are due to potentially a direct infection of those particular cell types by SARS-CoV-2. And as you know, ACE2 is a co-receptor for infection. I will include a caveat that I think a lot of folks have seen time and again, especially as you look at later time points. Oftentimes, what you find is that you see lower ACE2 levels in certain cell types, or those cell types end up being depleted, and that's presumably because if the infection is killing them off, you might not detect them as robustly as well. And so that's just something as you go in the later time points, the expression levels there might not be as reliable. And on the flip side, there are opposing effects where some people believe that ACE2 is an interferon-stimulated gene too, which might cause upregulation in the disease context.
Great. Dr. Philbin, let me come back to you with a question that came in during your presentation. And there's been a number of questions that have asked about how the data that you've generated might be used in the future. But this one specifically says that the hierarchical clustering that you showed shows two clusters. What is the future use of such clusters, and will these be used to develop predictors that can predict survival of patients?
Yeah, so this is one of the fundamental questions: how this how the plasma proteome can translate into clinical practice and how specifically our findings can. So one utility, is like we derived a classifier for severity and death that does seem predictive. The question, of course, is is a classifier that uses plasma proteins only, does it perform any better than the clinical parameters that we use when we're practicing? And that's an area that we haven't explored yet, but certainly does need to be explored. In the COVID positive versus negative model that we did, we saw a very clear distinction in proteome between the positive and negative patients, except for that small subgroup of patients, like I mentioned, that were COVID positive, yet did not demonstrate this pro-inflammatory signature at all. And this kind of leads to the thought of, well, classifying patients based on their plasma proteome signature and potentially triaging patients to certain therapies or not. As you can imagine, patients who don't mount the typical pro-inflammatory response might not respond well to glucocorticoid therapies. And Arnav can probably add to this, but we've explored different ways of classifying these patients or phenotyping them, so to speak, based on their plasma proteome. And the hierarchical clustering that we did is, of course, only one way. And we certainly want to dive deeper into this concept of patient phenotyping, not only predicting outcomes, but specifically predicting response to therapies.
Great. Dr. Mehta, did you want to add anything?
Yeah, I'll just add a quick comment that we didn't have time to show it today, but we have explored other approaches to clustering patients, including graphical clustering, and I think that's an area where we're actively pursuing and very interested in trying to understand patient endotypes.
Great. And just to confirm, all of this data is publicly available, including any tissue signatures that you developed, correct?
That is correct. So, graciously with olink, we were able to release the data several weeks ago, with the hopes that the community at large would be able to use it. And, we're very interested in everyone else's insights on what we can do with the data. But in addition, we posted an initial preprint on bioRxiv that has all of our supplemental tables, including the protein signatures as well. And obviously, feel free to email Dr. Philbin and myself if there are any questions or if there's any other data that we're happy to provide or help with.
Mm-hmm. Great. And Dr. Mehta, just another quick clarifying question that asks what proteomic platform was used for the prediction?
So the olink proteomic platform.
Great, thank you. Okay, another question. I'm going to come back to you, Dr. Philbin. So this is another prediction question. Could the plasma proteome signature be used to determine which patients are perhaps candidates for vaccines?
That's an interesting question. I would posit yes, and I would point to the findings of our neutralization assays, and the fact that there's quite a bit of variability in the development of the neutralizing antibodies in the setting of disease, and I can imagine that there's quite a bit of variability in response to a vaccine as well. And one thing that I think that this platform provides is kind of a baseline signature of how patients respond to disease and how patients variably respond to disease, which provides a great backdrop for not only studying potentially vaccines, but specific therapeutics. And what is really interesting, or what would be interesting, is the fact that we enroll patients so early on during the surge here in Boston, and the only therapy at the time was really there were no therapies at the time, and it was prior to remdesivir. So our patients are essentially receiving supportive care only, and to study populations of patients who are receiving certain therapies, or in association with clinical trials, this tool could be very powerful in kind of elucidating the differential expression and responses to both vaccines and therapies.
Okay, I'm going to come to Dr. Mehta now with I think a more mathematical question, so I think you'll probably like this one. In the analysis of COVID severity, there are hundreds of proteins that are significant. Have you looked at the distribution of the p-value, for example, via QQ plot, and is there any evidence of p-value inflation?
Yeah, so it's a great question, and it's something that came up right away when we were looking at the data as well. So one of the ways in which we approached this is that we did a permutation analysis where we randomly assign severity annotations to each patient, and we repeated this several times over, so 1,000 times over, and looked at the distribution of p-values and the number of significant proteins that one gets from those analyses. And it's very clear that in the true case, we are very far into the outlier. So it is very unlikely that we get this many significant proteins and this distribution of p-values that we get with our analysis than in a randomly generated set. So that gave us a little more confidence about some of the associations we're seeing. And the other thing that I think is important, and it was brought up earlier, is with any such data set, having a validation cohort is important as well. And so that's something that, we're actively working on to validate our findings in other data sets. And so we're excited to see some of the other olink data sets, being released where we can do this.
Great. So we are almost out of time, but I have a really nice question, I think, to finish off with. And I'll put this to both of you. Why don't we start with you, Dr. Philbin? What are some of the future plans that you have based on your findings from this study? Where is the research going to go next?
Well, the next step is to merge, our findings with the single cell sequencing of PBMCs that is currently ongoing at the Broad Institute of all 306 of our patients, which is really going to provide an amazing resource of data, and we'll make all of those data public as well. But really being able to, on a patient level, associate the plasma proteome with what's going on at the level of single cell RNA sequencing. So that's quite exciting. We're also going to receive soon data on viral load for these patients and at the different time points. So I think it's going to be very interesting correlating viral load with the signatures that we're seeing in the proteome. And I'll let Arnav cover the other 10 things that we're planning to do.
Thanks, Mike. I'll make 2 points generally, because I think the data set in of itself, what we're hoping to do with this is, one, be able to leverage it as a general resource. So first, to understand what is really contributing to the plasma proteome and how can we best dissect the relative contributions. And so I think it's very important, and we're very keen on understanding, obviously COVID-19 biology, but we anticipate this will be useful as a more general resource in other pathogenic states. And then the second point I'll bring up is that, we have a very large cohort here with very interesting proteomic data. And so the other thing that we are actively pursuing and have been thinking about is how can we leverage the data set and sort of the range of demographic and physiological variables that we have to understand other aspects of human biology outside of viral infection. Such as the aging and the host immune response. And so I think those are avenues that, we're very interested in and are looking into pursuing.
Oh, that's wonderful. But unfortunately, we have run out of time, so we're not going to be able to answer any more questions. But before we go, I did want to thank our speakers very much, Dr. Michael Philbin from Massachusetts General Hospital, and Dr. Arnav Mehta from the Broad Institute of MIT and Harvard. Please go along to the URL in the Resources tab to find more information about products and technologies related to today's discussion, and look out for more webinars from Science available at webinar.sciencemag.org. This particular webinar will be made available to view again as an on-demand presentation within about 48 hours from now. If you'd like to receive alerts about future webinars, please just go to the sign up link in the Resources tab to the right of the slide window. We're interested to know what you thought of today's webinar. Send us an email at the address now up in your slide viewer. That's webinar@aaas.org. Again, thank you so much to our fantastic panel and to Olink for their kind sponsorship of today's educational seminar. Goodbye.